didn't look at the words.
The hour has arrived. So welcome everyone. Thank you so much for being here. My name is Rebecca Terry. I am Jesse's advisor, and it's my distinct honor to introduce him to you all today. Gosh, 8 years. 8 years. So I first met Jesse in 2016 in the summer. He replied to a research assistant position that I had just posted because I needed help. I had an infant and a 4-year-old, and I was coming back from maternity leave, trying to like pick up the pieces and get things going again. And I got this 4-paragraph long email plus a very long CV from the person from the Fish and Wildlife undergrad at the time, who was clearly wildly overqualified for what I was looking for. Like so intimidating to get this CV on my desk, right? And somewhat pathetically, he reminded me the other day that he had emailed me from the Mount Airy National Wildlife Refuge, where he was an avian point count technician at the time, and also planning on after the classification of the refuge in 2016. And I say prophetically here because Oregon's Great Basin corner of the state was an area that I was very interested in, even before I had the job here at Oregon State. And I had several conversations with Doc Storm in 2013, 2014 about getting some research started in that corner of the Great Basin. But like I said, I had a 4-year-old and an infant, so that didn't have to be the case. So he arrived, he interviewed a month later. And I had no idea how much this person was about to change my life, you know, but I knew that I had just got really lucky that he was really competent, organized, and like perfect person in the lab. So I remember going home and telling Mark, I think I had just won the undergrad jackpot. And I had. So I quickly learned that there was a lot more to Jesse than just that very impressive CV, and that he had had a lot of life before becoming a student here. He hails from the middle of Tennessee, and he had been a photographer. He holds an associate's degree in photography. He had been a pizza chef or a pizza server, I don't know what you want to call it. He'd been in a rock and roll band and toured around, like actually professionally. So they're called the world's greatest ghosts. You can go Google them. And then he had gone back to school at Mountain Hood Community College and gotten an associate's degree there in natural resources technology. And then with the help of a Ford Family Restart Fellowship at OSU, and was getting a fish and wildlife degree. He also had a kid, similar age as mine. That kid is sitting right here in the audience. That kid was a lot smaller. So we started, so we started talking his graduation year, and he asked me about being a grad student, and I said no. Hoping that I wasn't making a huge mistake, feeling like I might be making a huge mistake, but I said no. Instead, I hired him as my lab manager for 9 months, give him time and space to really like breathe and think about what he wanted to do in grad school. And he had also just become a parent for the second time, and so there was a lot going on. So during that time, we started talking about this idea of a historical resurvey out on the Steens Mountain. And I remember Jesse telling me he was so excited by this idea that he couldn't sleep. And I kind of felt the same way. So I thought this was a sign, right? He's ready. We're ready to tackle this. And he was just the perfect combination of skills, connection, the grit to pull off a project of this scale. So he started with me as a PhD student in April of 2018. And as you're about to see, he has done Bob would say, bang up job of this work. He has poured a massive amount of intellectual and like physical work into this research, and he's taken this project to heights with his creativity and his drive that I could never have even imagined at the time. I'm just insanely proud of everything he's done. I'm thrilled he's reached the finish line. He has been hired by the Institute of Natural Resources at Portland State University and is going to become there's a Wallaby program manager at the Oregon Biodiversity Information Center, and they are about to find out if they hired. But truth be told, I've been actually kind of dreading this introduction. It's really hard for me to write it. So with that, I'll leave it there and just turn it over.
Thank you, Becca, for that wonderful introduction. And thank you all for being here. I'm so incredibly honored to see all of your faces today. Thanks to those that are tuning in on Zoom as well. So today I am going to discuss my dissertation research studying birds and mammals and their diversity across different spatiotemporal gradients, with a specific emphasis on the role that mountains play in driving that diversity and those patterns. And I thought we would start today with this image here. So this is Steens Mountain, viewed from the Alvord Desert. It's located in southeastern Oregon. And I'm going to be spending a lot of time talking about this mountain later on in my talk, but I want to start here because this is often how we think of mountains, right? So they're out there, they're in the blue, they're on the horizon of our collective consciousness, and they inspire our imagination. They call to us to come and play, to come and explore. But they're also incredibly important places for biodiversity. And because of that, they're these laboratories for us to understand the natural history of taxa and to think about their ecologies. And biogeographers and ecologists have been thinking about mountains and diversity for a long time. So this is Seahart and Miriam, who studied birds and mammals in North America. And Miriam described these biogeographic maps across the continent that were distinguishing big changes in plant communities and how those harbored different assemblages of animals. But he also thought about this along altitudinal gradients as well, establishing these different altitudinal life zones, which were changes in humidity and temperature that harbored different assemblages of taxa. And this is kind of the concept of an elevational diversity gradient. And this was an important idea because mountains harbor high amounts of biodiversity. And the reason for this is as you change elevation up a mountainside, there's differences in land cover along that mountainside as well. And those different land cover types are different habitats for different assemblages of faunas up the elevation gradient. So mountains really do disproportionately harbor much of the world's terrestrial biodiversity. They're really biodiversity hotspots. But how do we measure biodiversity? Well, as it turns out, biodiversity is multidimensional, and we can think of it in, yes, the taxonomic dimension, which I think we're all very familiar with. You can look at species in a community, you can quantify and count how many species are within that community using something like species richness within that community. There it goes. No, it's not going. Anyways, so you can count the number of species in a community. Maybe you think about like which species are there in that community. And that's an idea of species richness. It could include abundance information and think about like, well, how many of each one of those species might be present. But we can also think about other dimensions of biodiversity. For example, we might be interested in thinking about evolutionary diversity within a community, and that's the phylogenetic dimension. We can use tools like phylogenetic trees to quantify the amount of evolutionary diversity contained in a community. For example, we may have very closely related species, such as this crow and this jay, or we may have very distantly related species to those, like this wren here. And we can do things like add up branch lengths on a phylogenetic tree and then quantify the amount of evolutionary diversity within that community. But we can also consider a functional dimension as well. We can use species traits to do this, species traits that, for example, might describe diet. We can then plot species within this diet space And then we can use metrics like a functional richness to think about like how functionally diverse a community is in terms of the ecological function going on. So I'm going to use this multidimensional framework to think about biodiversity today in this talk. And we're going to explore this in both birds and mammals across different gradients in time and space as it relates to mountains. And I'm going to be doing this in 3 separate chapters of my dissertation today. The first chapter, I'll be taking a really broad scale approach and talking about the role that mountains play in multidimensional biodiversity of North America's songbirds. Then we're going to be zooming in to one single elevation range, where I'll be discussing a historical resurvey that we did conduct of small mammals in the Northern Great Basin to the tune of a century's worth of ecological change. Then finally, I'll zoom in even further and I'll discuss ecological niche breadth dynamics in 2 rodents along that same mountainside. But let's go ahead and we'll get started talking about the largest clade of birds and my personal favorite, passerines. So passerines are in the order Passeriformes. And I mean, they're just awesome, right? I love them. They're fun to count. They're fun to chase if you're a bird watcher. Passerines, like many birds and other groups of animals, have what is known as a latitudinal diversity gradient, where species richness increases as you move toward equatorial regions of the globe. But as you guessed it, I'm also going to be discussing patterns in biodiversity and passerines across elevation, so an altitudinal diversity gradient as well. So in this part of my talk, my objectives for this chapter were to map seasonal biodiversity patterns of all naturally occurring passerines across the continent in these multi-dimensional aspects of biodiversity. And in doing so, I hope to uncover these hotspots of multi-dimensional biodiversity in songbirds, and then assess the relative influence of topography, land cover, and climate. Why do we do this, or why did I choose passerines to do this? Well, aside from being inherently awesome, they're the most diverse group of birds. There's over 500 species in North America, not counting regions south of Mexico. They're well studied in terms of their phylogeny and their functional characteristics. They're roughly the same in size, except for a few outliers, but they're very diversified in terms of their habitat and their diet. There's a lot of conservation concern associated with passerines. Of 3.2 billion birds lost to North Americans' avifaunal abundance in the last, say, 50 years, roughly 90% of that is attributed to passerines. And I did this in North America because it's an incredibly topographically complex continent, and I'm interested in the role that mountains play. There's also this pre-existing network of North American bird conservation regions, which allow us to think about changes in diversity across different regions. The data sources I used for this study were species range maps from BirdLife International, which bring in that multidimensional aspect. I leveraged a global phylogeny of birds, as well as a comprehensive ecological database for the world's birds. And I also brought in environmental layers such as topography, land cover, and climate layers to ask questions: how does climate drive different patterns of diversity? The footprint for the study was the 66 bird conservation regions. And in the GIS, I overlaid this hexagonal grid cell assemblages at a 50-kilometer and 100-kilometer resolution. I'm only going to be talking about the 100-kilometer resolution for time today. Patterns are very similar. Calculate species richness. We brought in both breeding and non-breeding distributions of these different songbirds for all species across North America. We then clipped those to our grid cells, and then we simply count the number of species to determine species richness for every single assemblage across the continent. But for evolutionary diversity, what I did is I brought in a phylogeny, and I built a consensus tree using 1,000 different phylogenies. And then from that, we can count up the branch lengths within each one of those different grid cells to determine the amount of evolutionary diversity that might be contained therein. For functional diversity, we brought in different traits that tell us something about how different songbirds kind of make a living in their environment, how far they can move, what they eat, their body mass, and then that with our species themselves in the grid cells, we perform a principal coordinates analysis to determine kind of the functional volume in each one of those assemblages, calculating a metric called functional richness. That kind of draws a convex hull around the functional space within each assemblage. So doing this, then we get these three multidimensional biodiversity metrics that we apply to every single assemblage in our study area. But we also wanted to model this. We wanted to find out the potential drivers. So we brought in topographic, land cover, and climatic variables. Think about what might be driving these different patterns. But because we're using grid cell data, we have to deal with spatial dependency. And that brings in our friend, spatial autocorrelation, the modeler's best friend, right? So I'm not going to spend a whole lot of time talking about this, but the concept with spatial autocorrelation is you have a community, you have another community close by, and they're probably going to look pretty similar to each other, as opposed to something that's much further away, which is going to be rather different. This isn't a problem in and of itself, but it introduces a problem when you try to use kind of a linear regression framework, because you don't have the assumption of independence going on anymore. So you have to account for that in some way, which is what we tried to do here. I use simultaneous autoregressive regression models and a spatial weights matrix to kind of weight these communities that were closer together in space, and hopefully in doing so to account for spatial dependency in their error terms. Okay, so let's talk about the results of this study. So here we have our study area and our grid cells, and this is what passerine diversity looks like in the non-breeding season. And this is what we kind of expect if we know something about migrating songbirds. Yeah, most of the species richness in the winter is going to be down in the southerly part of the continent, the highlands of Mexico. When we kind of progress to the breeding season, and you can watch as warmer colors kind of creep up the screen here, we see that during the breeding season, though, this species richness is much more broadly distributed across the continent. And when we underlay a topographic hillshade, what we see is that many of the areas harboring such rich passerine species richness are mountainous areas. And these are some of North America's great mountain chains. For example, we have the Sierra Madres down in Mexico. Also, we have the Rocky Mountains, the Appalachian Mountains, the Sierra Nevada, the Cascades. And these are areas where we see a lot of species richness in songbirds during the North American breeding season. When we compare species richness with phylogenetic diversity, they pretty much mirror each other. We have a lot of congruency between those two, which isn't really surprising that mountains also harbor high assemblages with high levels of evolutionary diversity. But when we look at the functional dimension, we actually see there's mismatches in places. So we have incongruency. Particularly, we can see that functional richness here extends much further north at higher levels, all the way up into Alaska. One of the reasons that I hypothesize this might be going on, there's harsher conditions further north. So we may have less speciose assemblages, but more ecological diversity in the species that are there. There's also incongruency here in this area around the Great Lakes. And this is an area where we also see some high species richness, but this is an area where we see no large mountain chains. So what might be going on here? Well, we also need to consider land cover, right? So this is a kind of a mixed forest area where we have the eastern deciduous hardwood forest coming into contact with conifer forests that are coming down from the north, and this influence of the boreal forest further to the north, and a lot of very patchy, very heterogeneous environments, which harbor these very diverse and rich songbird communities in the breeding season, perhaps with more ecological function that we're seeing here. So using all of these data, I derived these biodiversity hotspot maps, which are the upper quintile values of each one of those multidimensional metrics. The red dots on your screen are 100-kilometer grid cells during the breeding season. The blue is the non-breeding season. And then those yellow grid cells are important for multidimensional diversity year-round. And then we quantified some of these bird conservation regions which have many of those grid cells. So these maps could be used for conservation purposes, but it would be important to take into account that these are only thinking about passerines here. Other birds are going to have other requirements, other conservation concerns. I was also interested in digging in a little further and thinking about not just passeriformes as a whole, but some families and what their dynamics are. So I chose 10 very speciose families within passeriformes, and I don't have time to talk about all of them today, but I'm just going to cover a few of my favorite highlights here. So several of the families, we actually see a strong latitudinal diversity. But we don't see that in all families. And in particular, Perulidae, which is the wood warbler family, we actually see a reverse latitudinal diversity gradient here, where we see higher species richness further north in the breeding season, really associated with this interesting mixed forest area, as well as in the Appalachian Mountains. And one of the reasons that has been hypothesized why this may be going on here is because of this productivity boom that happens within these forests early in the breeding season. And with this plant productivity comes a lot of insects. And these warblers, because they are insectivores, may have evolved in their migratory path. May have adapted through their migratory pathways over time to track these rich resources so that they can provision their young during the breeding season with them. There's some conservation implications here, right? So, we know environments are changing. If there's phenological mismatch between when, insects appear in the landscape and when birds are migrating, that's something to consider for conservation research. So when we think about the patterns we see across all of these families, we see that there's very different families because different families are very different patterns because different families of songbirds do very different things. And this plays out not just in species richness, but also in phylogenetic and functional dimensions with differing levels of congruency and incongruency depending on those different families. I'll show you one more example here that I think is neato. So these are finches, and finch species diversity actually peaks in the montane west here, both further north as well as down in Mexico. And I hypothesize that maybe one of the reasons why this might be happening has to do with peak conifer diversity. These areas are kind of global hotspots for conifers as well. And many finches are adapted for seeds of conifers and take advantage of that, as well as many other plants. So this is an idea I would like to continue to explore for future research. Okay, so we modeled this using those models that try to account for spatial dependency. And what we find is that species richness, phylogenetic diversity, and functional richness, yeah, so the amount of elevation relief is pretty important, as well as mixed forests. So we have to consider land cover and then some climatic variables as well, driving all of the diversity we see across the entire clade. In the non-breeding season, we see that elevation relief is still really important for all of these different biodiversity metrics. But temperature becomes less important. We see some interesting things like shrublands and not grasslands having a positive effect on passerine diversity during the non-breeding season. So I don't have time to show you all the different families and their modeling, but there's a lot of interesting things to dig into. So be happy to chat later about it. So in summary, what did I learn in this exploration? Well, yeah, mountains are hotspots of biodiversity and songbirds, but especially during the breeding season. Topographic relief, seasonal climatic variables, and mixed land cover types are really important for predicting this multidimensional biodiversity. But different songbird families with different ecologies and different evolutionary histories have different diversity patterns because they may be responding to different things across the landscape. Okay, so I'm going to zoom in now from all of North America to spend the rest of the talk talking about Skeens Mountain, one single elevation gradient. So this is a view of the Alvord Desert from the eastern rim of Skeens Mountain here. And I'm going to use this as a stage to explore diversity not among birds, but among small mammals. Yes, across space, but also across time, over about a century's worth of time. And I did this using a historical resurvey. So a historical resurvey leverages records from the past, things like old field notes, museum collections, natural history specimens, old reports. And we can use that information to think about how things have changed or not changed over time when we go back and we revisit these sites and apply modern techniques in field resurveys. And this has been done pretty effectively in other systems here in the Western United States. Very famously by UC Berkeley Museum of Vertebrate Zoology researchers when they went back and they resurveyed Joseph Grinnell and colleagues' work in California in a lot of different montane systems. But why? Why do a historical resurvey? Well, long-term monitoring is actually pretty rare. It's really cool when it happens when you have long-term sites, but they're kind of spatially restricted by design. And if we want to learn about how things have changed over time, this is an approach we can use. And we know that things are changing, right? So we're in the middle of a 6th mass extinction event on this planet. which is largely being driven by anthropogenic activities, changing the climate, and it's projected to become much more drastic depending on what we do and how fast we do it. And we alter habitats. And some of that alteration has been going on for a long time, from both direct anthropogenic effects and indirect effects, particularly here in the American West, as we see land cover change over time with woody encroachment. I study this with small mammals because small mammals are great ecological indicators. They're functionally diverse, they're really sensitive to changes in habitat and climate, and they connect primary plant producers with predators. So they're really trophically important. They can tell us a lot about a system. They're really bellwethers of ecological change, and we can study them over time. So let's take this like cartoon small mammal assemblage on this mountainside, and let's say over time, maybe climate warms and it gets warmer as well. Certain species that are adapted for certain conditions, maybe they can no longer persist and they start to blink out of certain elevation bits. Other species may shift their elevations up or down to accommodate changing conditions, and you may have new species come in. So that makes, mountainsides a really interesting place for this historical approach, and it has been done in other systems here in the West, such as Yosemite National Park, as well as in the Ruby Mountains in the Great Basin. So in this resurvey, I sought to ask what small mammals have undergone significant elevation shifts from the early 1900s. How does community diversity compare through time in both taxonomic and functional dimensions across this elevation gradient? And then also thinking about space. How do communities change, or how can we compare them in terms of the diversity across both a geographic and an environmental space along this gradient? Okay, so this is my study system here. This is Steens Mountain, and Alvord Desert is located here in the northern Great Basin ecoregion, right at the border of the Columbia Plateau. So it's an interesting biogeographic boundary where you have this mixing of different faunas there, this subtle mixing. So it is a large fault block mountain. It's just one mountain, often confused for many, and it runs 70 kilometers approximately north to south. So like many other major geographic features, Steens has been known by different names. And this mountain is located in the traditional Aboriginal homeland of the Northern Paiute people. In the summer of 1860, U.S. Major Enoch Steen forcibly pursued and removed a group of Northern Paiute from this elevation gradient, this mountain. So I would like to acknowledge the forcible removal and disenfranchisement of the Northern Paiute, not only from this mountain, but from the Great Basin in general, by using its traditional name in the Northern Paiute language, which is Tsitsieda, which translates to cold, cold mountain. And it really is a cold mountain. Snowpack persists on the top of this mountain far into the season when it's very dry around in the surrounding desert. So today, you will hear me refer to the mountain, probably as Steens Mountain a few times because it's on maps, but I did want to make this distinction. And the Northern Paiute descendants, such as members of the Burns Paiute tribe, they still use the mountain as well as the surrounding lands to this day. So let's zoom in to this elevation gradient and see why it's such an interesting place to study diversity. And there we go. Okay. University technology, gotta love it. Okay, so so this elevation gradient itself, it captures all these different transitions in elevation within these 5 major elevation zones of the gradient. In the Alvord Desert Basin, we have really xeric and dry conditions. And as you move up to the top of the mountain, you get these more mesic and alpine conditions. The desert basin is about 1,200 meters, and up at the top, you're just under 10,000 feet at the summit. So you can see it really captures a lot of extremes, from really hot and dry to very cool, wet, but harshness on both ends, and then everything in between, making it an interesting place to study diversity. And in terms of the land use out there, there's a lot of grazing, both on private and public land, mostly cattle these days. And then a lot of recreation. So Alvord Desert's a great place to go party, pick up after yourself. And a great place for hiking. There's a lot of wilderness area. So it is an area of conservation concern and enjoyment. But there was a lot of historic overgrazing on Steens Mountain, primarily by sheep. So this is a photo from 1901, and it was estimated that there were 182,500 sheep grazing at high elevation on Steens Mountain. And let me tell you, that is a lot of sheep. Potentially 450 to 1,000 sheep per square mile. And this is what Griffiths in 1901 calls a moderately grazed native pasture. I'd say that's pretty hammered. This is what he's calling a depleted range. I'd say it's absolutely denuded of all of the vegetation there. So all the bunch grasses are gone. And it's reasons here on Steens Mountain, as well as many other elevation gradients in the Great Basin, why we had the Taylor Grazing Act come in 1935. And that over time, we've seen those systems kind of bounce back. So this is a photograph by Hansen from 1943. We can see at high elevation, we do have vegetation returning, but the scars are still in the landscape. And you can kind of look at the growth of the bunch grass and how it's elevated on its tufts. We still see the evidence back in the 50s of that earlier grazing. You can still kind of see it today as well. And there could be indirect effects of that grazing. And I don't have time to go into all the hypotheses why we have seen the encroachment of this tree species, Western juniper, in the Great Basin, but there could be an indirect effect of grazing as well as changing fire regimes. And if you ignore the sweet fins on this Plymouth right here in this photograph from 1960, and we compare this photo with a photo taken in 2013, we can see this expansion of juniper on the landscape. If you look at this slope here, this is the opening of Little Blitzen Gorge on Steens Mountain. We can see that juniper has really encroached on this landscape. In fact, there's a lot of juniper management that happens on Steens to this day. And aside from land cover change, there's been warming in the system. We have seen a decrease in the amount of snowpack on Steens in the summer months. In the recent decades, we've had 2 to 10 day earlier melting of the snowpack on Steens Mountain. So climate change seems to be having an effect as well. So the amount of ecological change over time and the amount of ecological variation across this elevation gradient makes it an interesting place to conduct such a survey. But how does one do it? So, well, it ain't easy. You start by defining your study area. Then you spend weeks, months, and years trying to dig up historical records. You find old PhD dissertations from this very department here that were conducted on Steens Mountain in 1956. Very different methods back then, it turns out. You find old maps and you overlay them on your study site to determine where these surveys took place. You find old NSF reports that were advised by Robert Storm in this very department. And you try to piece together this puzzle of what work had been done when over time. Other reports from Eastern Oregon College at the time, old field notes from Annie Alexander and Louise Kellogg, some pioneering naturalists from NBZ who visited the Alvord Desert and collected small mammals in the 1930s. You leverage museum databases and you go and visit old dusty museum collections forgotten by everyone else that aren't in any database, and you find specimens. You come up with this historical data set over time by cold calling people and the generosity of folks who want to share their data with you. And then you have to go out and you have to find where this stuff was collected. Not quite an easy task, but you start the scouting mission here. You go out and you drag your friends down really shrubby conditions for miles and miles down steep canyons. And you find the sites eventually, and you know you're in the right place when you find the original traps that they left behind accidentally, in some cases then being used, well, for other purposes in the interim years. Target practice. So what I did is I took this historical data set and I kind of downsized this to something more manageable. And as it turns out, we actually have pretty good coverage, not just from 1 year till now, but throughout these different decadal bursts of surveying that had been done in this region by many different people. I then selected 26 locations to do as my resurvey sites, trying to match historical sites, but also filling in the gaps of land cover types that we missed over time. And I think that we achieved pretty good congruency between the historical data set and where we sampled to this day. And then the real fun comes, which is the actual field work. So Our small mammal localities were these circular areas of roughly 0.8 kilometers squared. And within each one of these localities, we laid our small mammal trap lines in these different habitat patches, taking care not to cross these ecotonal transitions in vegetation, so that we can really sample the different small mammals that are inside these different habitat patches. Within a particular trap line, we laid about 20 to 30 trap stations, and we used a combination of both lethal and non-lethal traps. And we did a lot of removal trapping, is where you actually take these animals out of the environment, create specimens out of them. And the reason we did this is twofold. One, we were hoping to really sample the true diversity. And when you remove individuals from a system, you increase your odds of getting a new individual, perhaps a new species coming in on trapping in a subsequent night. But also we wanted to contribute to museum collections. And all of our specimens that we collected are going to the Burke Museum of Natural History and Culture in Washington, or staying here at OSU. So we baited our traps with yummy peanut butter and oats. We had about 100 to 800 trap nights of effort per locality, depending on logistics. We sampled the habitat really thoroughly along these trap lines as well. So we used 1 meter square quadrats to collect percent cover along all of our trap lines that were then used in subsequent analyses. We systematically went through and we clipped all the plants through all of our small mammal trap lines. We pressed them in the field. They were later identified by experts here at the OSU Herbarium. And we collected insects so that we could do stable isotope analyses on them, which I'll talk about here in a little bit. Okay, so after you get your specimens, you need to take your standard measurements on them. You spend lots of fun hours with your friends, assigning species. You're creating specimens from these. We used a variety of preservation techniques in the field. We prepared voucher specimens as skeletons, as study skins, as full fluid preserved specimens. And it was actually a lot of fun doing that. And we got to train a lot of amazing interns and field assistants in that process. I want to talk about field work. So field work is hard, y'all. I mean, it's not easy. You know, you're out there in the Alvord Desert, and it's 105 degrees in the shade in the summer of 2021. You're trying to find any relaxation and respite you can. Some days you're up on the top of the mountain just freezing your butt off in cold fog. Everything's wet, the wind's blowing. You're camped out in the middle of nowhere for days and weeks on end. You're hiking to the middle of nowhere. You're dragging your friends along with you during all of this, and you're hoping not to fall off the side of a cliff while you're doing it. You're dodging lightning bolts. You're sucking in dust during dust storms. But, you know, it's really not all that bad. In fact, it's pretty. I mean, there's always good stuff to look forward to. There's always a nice cool breeze blowing through your hair. I mean, it's really quite refreshing. And there's all kind of amazing stuff to step on out there too. It's really fun stuff. And, you know, at the end of the day, you can always count on your trusty old field vehicle to see you through and get you home safely. So, but yeah, okay. Let's get back to methods here. All right, so we were interested in doing this comparison of range shift between historical specimens and our resurvey results. And so to do this, we plotted the elevation ranges of our historical small mammal occurrences from the past with our resurvey specimens, and we adjusted the species occurrences from our resurveys using multi-species occupancy models, using detection covariates, with the amount of effort that we use and the traps that we use, and occurrence covariance based on elevation and elevations squared term. And to understand the change in these assemblages over time, I modeled temporal beta diversity. And beta diversity is just the difference in diversity between a community in one place or time and another one. So that's all beta diversity is. And we modeled that using distance decay. And we temporally bin our populations within these 5 elevation zones. So we had elevation zones with temporally bin communities therein. And then we took our mix of historical and resurvey data, and we plotted the dissimilarity over time in each one of these communities within each one of these elevation bins over time. And that's our taxonomic distance decay, thinking about how things have just changed in terms of the species that are there. But I was interested in also doing this functionally. So we brought in a suite of ecological traits that tell us something about how small mammals make a living in their environments, with our species occurrence data, performing a principal coordinates analysis, and then we can do a dissimilarity over time, but not in the taxonomic dimension, the functional dimension. And this gives us our functional distance decay over time. And we wanted to know how climate was also changing over the same time period. So we brought in these historical climate layers using PRISM data, and then we plotted climate by year and regressed those climate variables against the year. And then finally, I was also interested in what are the spatial dynamics of beta diversity in this system. So using just kind of geographic information, I estimated a geographic Euclidean distance using latitude, longitude, and elevation. And then I brought in habitat information that we collected in the field, deriving a structural complexity index from our trapline surveys, as well as climate layers and information about topographic roughness of these sites from remotely sensed data, and estimated the standardized multivariate environmental distance. And then using these two distances here, we can plot the dissimilarity of rodent communities just using our modern resurvey data in these two spaces. Then I broke these two down, this beta diversity down, into the two components that drives change between communities. That's one being turnover, which is really where species are replaced by different species in communities, or nestedness, where different communities are just subsets of the same species in different configurations. Okay, so let's talk about the results from this resurvey. We found 23 species in this system, over 10,500 trap nights worth of effort. And we collected 789 voucher specimens that are all going to museums. 3 of those were rodents, or 3 of those were shrews rather, and 20 of them were rodents. These are all photos that we took in the field as well. So we did detect some species range shifts, and I'm going to show you a figure and orient you to it, and then we're going to dig in a little bit. So when it loads. Okay, so on the x-axis here, we have elevation running from low elevation to high elevation. And on the y-axis here, we have all of our species plotted. The different color boxes are different taxonomic families for these taxa. And then the dark bars here, the solid bars, are indicating where we found these small mammals during our resurveys, where we trapped them, with the black dots being where we actually trapped them on the landscape in localities. And the red dots are where our occupancy models told us they should be, even though we didn't find them there. The dashed lines are the historical ranges along elevation of all of these different taxa. And what we found is we found some range contractions over time in certain taxa, a possible extirpation. We also found a range expansion. And there were some other species that I'm not willing to even call a possible extirpation because I don't think we sampled well enough for some of the small shrews in the system. I think I would like to go back and do it again. But let's talk about some case studies here. So this is the American water shrew. It's this amazing animal that lives in these fast-moving, cold mountain streams or in the periphery in the riparian area, hunting invertebrates. And what we found in our resurveys is that we see this upslope shift of 184 meters from its historical distribution. And this has actually been seen in other mountain ranges in the West that have done resurveys. So this is a very mesic adapted species, and it could be responding to warmer conditions over time in the system. We also see some other mesic adapted species such as long-tailed vole and montane vole, relatively mesic species that have shifted upslope as well from their historic distributions, 309 meters and 230 meters. Another vole species that we really tried to find and couldn't was the sagebrush vole. And we searched at many sites where it was represented historically, but we were unable to find it in any of our modern resurveys. I'm not quite ready to call this an extirpation because vole populations are prone to boom and bust cycles, but I do think it warrants some more monitoring in the system to really determine its status. We did find an expansion in one species, and that would be the pinion mouse, or Peromyscus trui, where in this system, we actually see this 900-meter upslope kind of expansion in its range. In fact, we think this might be a possible new colonizer to this system. So in our database, because it's restricted, the oldest records actually go back to 2007, although I know it was also found in the 90s regionally nearby Steens Mountain, but that's still pretty new on the scene. And one of the reasons we think this may be happening is because of a change in land cover over time. So remember, we have Western juniper that has really encroached upon this system, and the preferred habitat type of pinyon mouse or pinyon juniper woodlands. And this is a story that's played out across the Great Basin in other ranges that have found that pinyon mouse has become a new colonizer and it has expanded its range based on this change in land cover. And land cover might have something to do with one other range contraction I'll talk about, and this is the least chipmunk, Neotamius minimus. And it was really widely distributed along this elevation gradient and trapped historically all throughout the Alvord Desert Basin. We didn't find any least chipmunk in the Alvord Desert Basin. We saw no chipmunks down low, and we only found 4 in really high elevations on Steens Mountain. So its congener in the system, the yellow pine chipmunk, is known to kind of outcompete it in woodlands. And we did find lots of yellow pine chipmunk. And with the expansion of woodlands, that could be part of what's going on. But furthermore, chipmunks across the West are somewhat destabilized. They're really prone to disease, plague, for example. And while we have no evidence of bubonic plague in this part of Eastern Oregon, it is known to occur. So that could be what's going on. And I think it is a species that warrants future monitoring in the system to understand what's really going on. And is it really gone? So let's look at climate over time quickly here. We did see significant warming of minimum temperatures on Steens Mountain over the last century. And that's attributing probably to that, that earlier snowmelt every year. There was also a mid-century peak in precipitation, although I don't know that has anything to do with the dynamics we're seeing playing out with the species. So yes, there has been warming over time, but in terms of overall community stability, we actually see a lot of that. So these communities have remained relatively stable across these different elevation zones. And we only find like a statistical difference in terms of dissimilarity over time in the Alvord Basin. And only in the taxonomic dimension. Functionally, we don't find that at all. So yes, there has been change in land cover and climate. Certain species are responding, but there's been some stability so far. In terms of the spatial beta diversity that I was interested in gauging, we see dissimilarity increases both taxonomically and functionally across space, which yeah, these are very different communities as you move along the elevation gradient. In the taxonomic dimension, it's really being driven by turnover, so different species. But functionally, we see not a strong signal in either turnover or nestedness. So that leads me to believe that, where species are changing, functional diversity might be changing a bit more gradually, where species are doing similar things, even though different species, different communities. And we see the same pattern in this environmental multivariate space. In fact, a tighter relationship is what our models are telling us, which, yeah, this system captures this environmental gradient really well across space. Okay, so what have we learned quickly here? Running out of time. We've learned that small mammal communities in aggregate have remained stable over the last century, despite this increased warming and temperatures. But some individual species have shifted ranges over time, perhaps due to that juniper encroachment or a warming climate or both. And even though species turnover is strong across changes in space, ecological function may change more gradually. Okay, so I'll spend a little bit of time talking about a third chapter here, staying on the same elevation range. And I'm going to be discussing niche breadth dynamics in 2 different species of rodent here on this mountain. And when I say the niche, so the niche is the range of conditions in which taxa can persist and the resources they exploit. And we can quantify the niche using niche breadth as kind of an estimate of the volume of an organism's niche. Where generalist species, they have broad niches, right? They eat everything, they live everywhere in a system. But specialist species have more restricted or narrow niches. They're more specialized in certain resources or in certain habitat types. It's been hypothesized that ecological niche breadth is correlated with environmental heterogeneity and competition within communities. And that generalist species tend to thrive in very heterogeneous environments that have lots of diverse resources, but low levels of competition, as to where specialists tend to do really well in more homogeneous environments with more evenly distributed resources. But they can often outcompete their generalist rivals because they are so specialized in certain habitats or on certain resources. So leveraging these hypotheses, I sought to ask the questions, how does niche breadth vary among populations of specialist versus generalist rodents across this elevation gradient? I asked, what are the relative influences of heterogeneity in the habitat and interspecific competition? And then what potential dietary resources might be contributing to any variation in niche breadth that we find across elevation. I want to give a big shout out to my field assistant, Chris Young, for helping me to generate these questions sitting around a campfire. Okay, so our study species in the system that we focused on were Peromyscus maniculatus, which is the North American deer mouse. It's cresseted, so it is a habitat generalist and a dietary generalist. So it lives everywhere in the system and it eats, we think, just about everything. And then our specialist is Paragnathus mellifilosus. It's a heteromyid rodent. It is known to be more of a habitat specialist, more associated with sagebrush. And it's a presumed dietary specialist relative to our generalist here in that it's more of a granivore. Both of these species persist up and down this elevation range from the Alvord Desert to the very top of the mountain, making them good candidates to explore niche breadth. And for the rest of the talk, I'm going to reduce these cute, cuddly, furry creatures down into chemicals, and we're going to use the tool of stable isotopes to talk about ecological niche breadth. Where we're using stable isotopes of carbon and nitrogen as a proxy for dietary niche breadth, where we use the nitrogen signal as a relative trophic position between herbivory and carnivory, and carbon is reflecting the photosynthetic pathway of the primary plant producers that these consumers may consume. We can take tissues from these animals, we can estimate their isotopic signature, we can plot those in this isotopic space, say a population of these organisms, and then we can estimate the volume of their niche breadth using things like ellipse volumes themselves, where a bigger ellipse represents a greater niche breadth and a smaller niche is a more constricted niche breadth. So we got our stable isotopes from hair samples from both of these species, as well as from plants and insects. So we wanted to know how potential diet sources were reflecting this. We outsourced our isotope analyses to these different institutions. And then we modeled the results. And what we wanted to do is we wanted to group these populations by elevation zones along this elevation gradient. So within a particular elevation zone, we've got different localities with individuals coming from those different localities. And then we take those and we then plot their isotopic signals from their hair tissues into this isotopic space. And we estimate a population level isotopic niche breadth using SEAV, a standard ellipse area in a Bayesian process. We also estimated the range in delta 13 carbon ratios and the range in delta 15 nitrogen ratios to understand the kind of the range of isotopic signal in both of them. Okay, we modeled niche breadth relationships by habitat by calculating a structural complexity index using our trapline data. And then for other covariates, we brought in our plant data to think about the amount of plant general richness that might be contributing or having an influence on niche. And then we used isotopes from the plants in these elevation zones as well. Competition is really hard to measure in these type of communities. So we're using a proxy for the potential for competition, which we're using a metric called functional divergence. And we used abundance data from our field surveys, as well as ecological information about small mammals, performing, again, the principal coordinates analysis. And then we derived this metric called functional divergence. And functional divergence, we can think of the functional space in a community, and where we have abundances toward the extremes of that functional space, then maybe the critters therein are doing rather different things, and we have a less potential for competition, as opposed to low functional divergence, where we have most of the abundances centered on that functional space and potentially higher levels of competition. We wanted to understand how this competition from the community was impacting potentially these populations. We modeled this. We had to get creative because we have a mix of individual level predictors and group level predictors, but only group level response variables. So we use micro, macro, multi-level models, which is kind of a latent variable structural equation approach to model this, with our outcomes being our niche breadth. And finally, I wanted to know, what were the potential contributors from the diet pool that were available. So using stable isotopes from plants and bugs that we collected along these trap lines, corrected using trophic discrimination factors, we then plotted those in an isospace and then use isotopic mixing models to kind of estimate if these consumers could be eating these different potential dietary resources. So quickly, I'm going to go over the results here. And on the x-axis, we have delta 13 carbon. On the y-axis, delta 15 nitrogen. We'll start in our low elevation population, and we can see, indicated by these colors for the two different species, we have a lot of niche overlap in both of these consumers. And actually pretty broad isotopic niches in the nitrogen dimension here, but a lot of overlap. As we move up in elevation, what we find is we have some divergence now in their niche breadth. They start to pull apart a little bit, still quite a bit of overlap, depending on which ellipse size you go by. These are 40% and 95% standard ellipse areas. When we move up into our mid elevations, where you have really complex shrubby and lots of tree plant communities, we actually see a contraction of niche breadth. Some of this could be due to small sample sizes, particularly with our specialist species, which we didn't have many within that elevation band. As we move up in elevation, we see them broadening out a little bit more, all the way to the top of the mountain, but still a bit of overlap in these populations. This figure is showing you just what I showed you before, but more in a summarized version, with these gray bars being 95, 75, and 50% credibility intervals, and the black circle being the posterior modal distribution representing standard ellipse area from our model outputs. And what we can see is that within the desert basin, we have elevated niche breadth in both taxa. And then at mid-elevation, we see that contraction in niche breadth size. But we do see across all elevation, no real clear winner in terms of niche breadth between our generalists and our presumed specialists. We kind of see them swapping places in different populations and different elevations. We get no significant effect of either the range in carbon or nitrogen from the tissues of our consumers across elevation. We do see some interesting things going on with our covariates, such as a mid-elevation peak in structural complexity, which we would kind of expect in temperate mountains. And we do have a positive relationship with functional divergence, which tells me that competition is probably higher at lower elevation in these heteromyid-rich desert communities versus the top of the mountain. But what we don't find in the modeling that we use are any significant relationships with niche breadth as measured by SEAB in any of our covariates. In fact, the only thing that our models show us is that we do have some significant relationships in the range of delta 15 nitrogen in the tissues of these animals with the range of isotopes of carbon and nitrogen within the plants and some of our trapline predictors. So an interesting thing that we didn't expect to find was that we had elevated niche breadths in the nitrogen dimension within the desert basin. But the more we thought about this and the more I read, it turns out that we find a lot of enriched nitrogen in very arid desert systems, particularly at low elevation. So part of what may be driving this variation across this gradient could be that we have geologic processes contributing to the spike in nitrogen, maybe physiology with these desert adapted plants or microbial soil crusts. And this is an area that I would like to continue to explore into the future, and something that we've talked about doing here in our lab group. Finally, just to look at the mixing model results in terms of potential diet items here. These are different elevation zones with our putative diet estimates of what these things might be eating. And we can see that there are a few things that jump out in the desert basin zone here. Based on our sampling, it looks like they're eating more forbs than anything else. We do have our specialists creeping up a little bit beyond our generalists, but still quite a bit of overlap, a lot of overlap in these other categories. We do see in the aspen zone, they're eating a mix of forbs and woody shrubs. And then beetles are really kind of, according to these model outputs, something that our generalist species tends to be behind. Okay, to summarize this chapter here, we found overall similarity in isotopic niche breadth between Paragnathus millipullosus and Paramyscus maniculatus populations within most elevation zones. So in terms of the investigation that we threw at this, maybe it's not as specialized as we thought. No significant influence of any of the covariates in our modeling on isotopic niche breadth. And it's a reminder that fluctuations in isotopic niche may not simply stem from diet, but we need to consider the underlying variation in the isotopic baseline. And some of the things I talked about earlier are reasons why we might see that spike of nitrogen down low. And we should look at that in the future. Okay, so I've talked about a lot, 3 very different chapters. And now I'm going to summarize that in one little slide. But I explored how different dimensions of biodiversity are arrayed across gradients of photography, habitat, and climate in both birds and mammals. And I do think that my dissertation work contributes to our understanding of terrestrial ecology of desert montane small mammals in the Great Basin, as well as the greater macroecology of songbirds in mountains across North America. And I think there's a lot of work to continue in all of those areas. And I believe that the Steens Mountain Survey Project is a comprehensive benchmark upon which future research can build. In fact, members in our lab are doing that right now. My colleague Alyssa Submersion is using specimens that we collect in the field to ask questions about physiology and ecology in these animals. Other members of our lab are interested in exploring isotopes using this data, like Sean Hickson, and I would love to get back out there and do more work when I can as the past. Okay. Okay, so a dissertation is a mountain of work, pun intended. And I could not have done this work without the support of so many people, so many people. Many of those are here today. Many are watching on Zoom. And I need to thank this department from the bottom of my heart. I need to thank the administrative staff for their help, Tara, Teresa, Tracy, Trudy, Melissa, for helping me with all these logistics. I want to thank Bob Mason and Adam Schwinnard for helping to make me a better teacher in vertebrate biology, giving me tough love when I needed it, and giving me some freedom to explore how to teach. I want to thank my graduate committee. Doug, I have known you for many years. You've been a friend to me and a mentor. Let's go chase some rare birds together. Becca Rowe, who is joining us on Zoom from New Hampshire. She's been an inspiration to me for many years in the historical resurvey work that she's done. I really look forward to talking with her about this research more. Mark Novak, you've been just this great resource to me in becoming a better scientist, a better analytical thinker. Thank you so much for your help. You're an awesome guy and you're a great dad. And Tiff Garcia, I asked you to be my GCR because I think you're one of the coolest people here at OSU and I love your work. And it's just great that you're here. And Becca, you have been so much more than an advisor to me, but boy, have you been an amazing advisor. You have taught me so much throughout the years as an undergraduate and then as your graduate student. You're a great leader, and by watching you, I have become a better leader myself and a better teacher and a better scholar and scientist. You're a brilliant scientist, you're creative, you're a great collaborator. You're great to your students, you care, you're compassionate, and I look forward to continuing to collaborate with you in the future. Thank you for making me a part of your lab group, taking a chance on an old dog like me. And, thank you for making part of your family. Your family is a beautiful, awesome family, and I love all you guys. There are so many other people I need to thank for their help during the Steens Resurvey project. A mix of undergraduates and graduate students, so many undergraduate mentees throughout the years that helped me in some really hard conditions out in the field for weeks on end. I see some of you smiling out there. I could not have done this without all of you, and you should all feel as proud of your work as I feel as proud to have had you be a part of it. This work is your work too, and even though like everyone was just amazing in the field and in the lab and in the museum, there's a couple of people I really need to thank. Bridget Reagan stuck by my side during a really hard field season. And she was just super tenacious, asked amazing questions. And I cannot thank Bridget enough for her help. Bridget went on to help me with some GIS work afterwards in an internship. So thank you very much, Bridget, for your help on this. I look forward to talking to you more soon about publications. There's another individual who I'm not sure if he's here. I have to thank Chris Young. So Chris Young has been my right-hand man through a lot of this work. 3 field seasons, helping me to come up with one of the chapters of this dissertation. I have watched Chris become a better scholar, a teacher now, and a father. And when things get hard and difficult, and push really comes to shove, Chris knows how to get out and push, literally, and get us out of a box canyon that's filling up with stormwater. And then, he just kind of like sits back and starts reading. So yeah, if this is a field biologist, I don't know who that is. I gotta thank my community. Like many of you are here today or joining online, all of my friends, my Portland peeps, my bird watcher friends, my D&D Zoom buddies. I couldn't be who I am without all of you. My friends near and far, old and young. Thank you so much. You're my family. I love you all. I can't say enough to thank you. And I want to thank my family too for all their support. I want to thank my parents who are online right now, who will be joining me tomorrow to celebrate, hopefully. And I want to thank my in-laws who are here today, Phil and Jeannie. Everyone thank Jeannie for the amazing treats that she made that are sitting outside there. They're so good. I want to thank my brother Casey, who's here today. I love you, man. Glad you're here. Megan and Mike, all these amazing people in my life. There's two very special people that I need to thank who are here today, and those boys. Jasper and Lark, you are two of the most creative souls I've ever met. You're amazing kids. I love you so much. I am so excited to see what the future holds for you. And whether that means being a marine biologist or a professional chef or a YouTube influencer or a clarinet player, whatever path that either of you choose, I'm going to support you 100% all the way. And yeah, you're great children, great guys, really good Lego builders too. And then finally, I couldn't have done this without you. Anyone that knows me knows that. Like your support through this has been huge, but beyond just supporting me, you're my best friend. And you have come with me on so many different adventures, fleeing from hurricanes to move across the country in an old beat up van, to touring in a rock band with me. I mean, look, she's a rock star. Coming with me on adventures, going through the adventure of being parents together. You're my love in life. You're my muse. You're my adventure buddy. Let's go have some more adventures together. But I want you to choose the next adventure. It's been a long one. So that's my talk. If I have time, I know I've gone over. I'd be happy to answer any questions. Thank you so much all for being here. Thanks.
We're slightly over, if people need to run away, I just want to say that assuming that all goes well, the closed door session, we'll be having some after party treats over in CRV, probably starting around 1 o'clock. Everyone is invited. Now, if you have any questions, I would say you can take a few.
Yeah.
Hi. First of all, phenomenal work. Really, really great. Second of all, I love that you acknowledge the fight. That's really good. Third, I am super excited that you're going to be working with us.
I am too.
It's going to be really fun. Fourth, the fact that you use the word veto. Rock my soul. That is really great. But my question is what would you like land managers take away from your research? Any aspect of your research?
It's a great question. And it's one that I could probably spend a long time talking about. But I think it depends on scale, right? So I think a lot about scale. And these investigations all took place at different scales. So the inferences that we draw from them, we need to take scale into account. So if a land manager is focused on a single elevation range, then we want to think about how conditions may have changed in that unit area versus if we're talking about big ecoregions, how change occurs at that. And we want to think about our objectives really clearly and what we can achieve and what type of tools and investigation that we need to bring that's appropriate for that scale. And I think that that's really important for land managers to think about. What tools and what data are appropriate for scale to answer questions. And I think that's one of the things that I really tend to think about a lot. Hey, Jamie.
That was great, Jesse. It's really cool to see all your work all put together like this.
Thanks.
I had a question about your diversity models and the fact that I believe I saw that temperature was kind of having no predictive value for diversity. And I was curious if your model could detect if that was a U-shaped relationship, because I think you would predict that the extremes would select for lower diversity, but maybe not a lot of.
And this is talking about the pass run work toward the beginning of the talk. Yeah. So I just showed you the results thinking about passeriformes as a whole during breeding and non-breeding seasons, but we also modeled this in different families as well. And I didn't show that today, but we do see temperature variables being important for some of those different families. But in the non-breeding season, yeah, we saw that temperature becomes less important for kind of this diversity and aggregate across the whole clade. Whether that's a U-shaped kind of pattern, I'm not sure. I do think that a lot of the reason that we're seeing temperature perhaps not being so important is because much of the diversity pattern at that stage during the non-breeding season is all down centered together in lower latitudes. So that might have something to do with it. And that when it's more broadly distributed across the continent, temperature maybe is more important because there's more variation in temperature during the breeding season. I don't know, I'd be really interested to talk to you more about that.
Yeah.
Part of
It's a question that says, what do you think is the future importance maintaining and continuing to build museum collections?
Yeah, I think it's incredibly important. I think that, so the types of questions that I ask leveraging museum collections just scratches the very surface of what you can ask of those. So most of my museum work, I visited a few regional museums, but I got a lot of that from museums that were published online, so I had access to those data. But if you go and visit museum collections, you can measure specimens, you can use morphometrics, you can do genetic analyses. And if we don't maintain these types of collections, and if we don't make them accessible to people, then they're not serving their purpose. So, naturalists in the past went out and sacrificed these animals so that we could use them. And we've done that here on this system so that future folks can use them. And so hopefully they'll be able to do surveys in a different way than we did them here and not have to remove animals from the landscape. But if we're not using them the way they need to be used, then I believe it's a shame. And I think they're incredibly important. So departments and universities, institutions need to be funding these collections. They need to be staffing these collections with curators, with managers. It's really important. Thanks, everyone. Appreciate you being here.
How are you?